Now the standard analysis tools for this systems biology are actually large data sets. And another very interesting aspect of the systems biology as compared to other conventional biology is that in the regular way in which we do biological sciences, we actually set up experiments and then we get data sets. And after getting the data set, we actually sit on the data and try to analyze it and make a conclusion out of that data, right? But in systems biology, the approach is a little different. Here, we start with a large data set, and then from that data set, we try to find out patterns and try to find out networks and try to find out what exactly that data means to us. So it's a little different approach. So rather than starting from data sets, rather than starting from experiments and going to the data set, systems biology actually interrogates a large data set and trying to find out the hidden information in that particular data sets, in the terms of the patterns, in the terms of this information content and so forth. So the systems biology is actually, as I mentioned in the previous, just a few minutes back, that it actually helps us to understand the systems as a total. So I will give an example. Suppose you take liver cells. So you take a small mass of the liver, a small tissue of the liver, and you grow them as a small organotropic culture, right? And then some other experimenter is trying to do a similar kind of studies, but instead of taking the whole liver tissue, culture those hepatocytes and do his experiments. So there are two different people. One is studying the whole liver, a small part of the liver as a tissue in an organ. Another one is going to disperse that into smaller tissues. And if they do the same experiments on these two different systems, the experimental result is going to be way different. The reason is that when you disintegrate in complex tissue into smaller components and try to do an individual based experiments and then try to convolute it back to the conclusion, then it actually sometimes becomes difficult than what the information will be or what the results will be from the whole tissue. Because it is very hard that you join the pieces together and reach to the same conclusions than if you analyze the whole thing together. And that is why exactly the systems biology is so important, that rather than doing a reductionist point of view where it takes a complex systems and then breaks down into simpler components and then getting that information and trying to integrate that, systems biology starts with a complex system and it assumes that the systems are complex, and then we are going to study everything together. And it assumes that the systems are complex, and then we are going to study everything together. So in a systems biology kind of an approach, you need not again has to take a complex tissue per se. So I just want to clarify that you don't have to start with an organ, you don't have to start with a big large tissue. We can do systems biology even into small cells or single cells. The only thing is that systems biology is more of an integrated approach, where we just don't look into one kind of data, rather we actually look into the different types of the data that could actually possibly be integrated together to reach a very meaningful conclusions. I'll just give an example. Say for instance, you just look into an RNA expression data and then try to derive a whole lot of biology out of it. That's not going to work completely, right? Because unless and until you look into the protein profiles, unless and until you look into the phenotype of the cells and then integrate everything together, you're not going to make too much of a conclusion out of it just by looking into the expression array data. So this approach is actually a systems approach, a systems biology integrated approach. So you didn't have to start with a big large chunk of tissue to do systems biology. That's not the purpose. The systems biology is just kind of how you look into the data, how you design your experiments, and how you integrate the data from the different avenues and integrate together into one whole component and then make a meaningful interpretation of that particular data. So this approach is actually a systems approach, a systems biology integrated approach. So you didn't have to start with a big large chunk of tissue to do systems biology. That's not the purpose. The systems biology is just kind of how you look into the data, how you design your experiments, and how you integrate the data from the different avenues and integrate together into one whole component and then make a meaningful interpretation of that particular data. Now if you look into an expression array data sets, you see that there are tons of expressions of the different genes. The genes going, some of the few gene expressions are going up, some of the expressions are going down, and then we are trying to get an idea that what exactly has been happening to the systems. Now for a couple of genes, a handful of 4, 5, 10 genes, it is easy for us to do that manually because you just can look into it, just eyeball them, and then you can actually categorize them, that which sets of genes are going up, and which sets of genes are going down, or even which sets of genes are not even altered. Even that is also important. Very important to interpret the data. So again, if the data is simple, it's okay to do an individual approach. But if the data set is large, which by and large most of the recent and experiments which we do and most of the modern biology taking a large data sets, it may be an expression array data set, even mass spectrometry data sets, but those data sets will be so large that individually you cannot look into them, but you need algorithms and softwares to help us to analyze the data and find patterns out of it. So this is just an expression, log fold change expression of particular genes. So there is a fancy formula for that. It's not very hard for us to know that which particular gene has been upregulated or downregulated. So we just express them as log two fold changes and then try to find out with respect to a control or an unstimulated cells what exactly is the change in the fold expressions in our treatment cohorts, right? So we have an untreated cohorts, we have a treated cohorts, and we do measure the gene expression profiles in both of these two. And we take gene by gene and then we actually find out what is the difference in the expression between these two. If the expression is more in this treated cohort, then we say that the gene is upregulated, and vice versa. If it is actually less, then we say downregulated. And if there is not much change, significant change, then we say it's an unaltered things. So this is a standard formula which people most of the time actually uses in gene expressions microarray based studies. Remember, these formulas are mostly for microarray based gene expression. We are not even talking much about RNA-seq kind of a database, which needs much more smarter and much more different, but a little complicated algorithms to integrate the difference. So this is just an expression, log fold change expression of particular genes. So there is a fancy formula for that. It's not very hard for us to know that which particular gene has been upregulated or downregulated. We just express them as log two fold changes and then try to find out with respect to a control or an unstimulated cells what exactly is the change in the fold expressions in our treatment cohorts, right? So we have an untreated cohorts, we have a treated cohorts, and we do measure the gene expression profiles in both of these two. And we take gene by gene and then we actually find out what is the difference in the expression between these two. If the expression is more in this treated cohort, then we say that the gene is upregulated, and vice versa. If it is actually less, then we say downregulated. And if there is not much change, significant change, then we say it's an unaltered things. So this is a standard formula which people most of the time actually uses in gene expressions microarray based studies. Remember, these formulas are mostly for microarray based gene expression. We are not even talking much about RNA-seq kind of a database, which needs much more smarter and much more different, but a little complicated algorithms to integrate the difference. So this is again a heat map. So heat map is another very wonderful way to interpret the data, where rather than looking into individual genes, of course you have an information about the individual genes, but at a glance you can actually look into the pattern of how the data looks like, the whole pattern in one glance. That's the usefulness of this kind of heat maps, where just look into the data and then you could see what kind of changes in the color. So each of these data sets actually will be interpreted with the help of definite color codes. Remember, these color codes are very empirical, so different researchers put their own color code. Sometimes high expression is represented by red color, and sometimes low expression is actually represented by a blue color. And then you have a log to fold scale, which kind of tells you as in the legend that what kind of fold change expression we are actually trying to see. You could see in this particular data set, say for instance, if you say that blue is actually downregulation, you could see a few sets of genes actually downregulated, and yellow, if it is been upregulation, then you see another sets of the genes actually been upregulated. So this kind of fluctuation actually happens whenever we treat a system with certain stimulants, certain kind of an external agents like cytokines, hormones, or with different drug treatments. And it can be the tissues from a normal individual or from a cancerous individual or individual with cancers. We're trying to compare the gene expression profile of these two tissue sets and trying to understand and ask a very simple questions. How does the transcriptome of a cancer individual or individual suffering from cancer, any type of cancer, is different from a normal individual? So this is pretty much the conditions, and then that's what exactly I was trying to explain in the previous slides, where you have a color code and then you have a profile of, trying to categorize the gene from its high expression and the low expression values. A few of the genes, as I always mentioned, will not be altered. In fact, majority of the data sets will not be altered. You have small clusters of a small slice of those genes which will be differentially expressed. We use the term differentially expressed because they are differentially present in these two different populations, the treated versus untreated, the control versus cancerous one, or disease versus or normal cohorts. But there are some data sets which are way different from the rest of the data and we call them as an outliers. So most of the time we are actually not too much interested about the outliers, because as you appreciate and as you might have learned earlier, that this outliers tends to alter the mean or the average and is going to alter our interpretation. So you kind of keep these outliers separated for now. But again, remember, just because they are outliers doesn't mean that they are useless or they are junk information. Sometimes extremely important and novel findings could be hidden in those outliers. Only the thing is that it is not good practice to mix the outliers with the common populations, because that is going to have a lot of fluctuations. So you need to analyze them separately and not mix with the general data set, where you are actually incorporating a little more of variations. You are allowing a little more of variations to come in. And if you are going for 3 standard deviations, you are even more relaxed that we are considering a lot of different data sets which is different from the average and so forth. So as the deviation actually increases, you are actually allowing a more number of data to enter into the data sets for analysis. But if you are extremely sensitive to some kind of alterations, then we are going to restrict our analysis to 1 standard deviation. That is approximately about 65 to 66% of the data sets, but that actually makes the conditions extremely stringent, and we actually say that okay, that distribution is very tight because we actually excluded majority of the data and we just took the data which is actually within that particular defined location. So this is actually again an example of how the 2 data sets might look like. If you see the figure on the left hand side, that actually shows a very little scattering. You could see majority of the data is actually sitting very tightly on that particular standard deviation, very tightly on that average. So there is a very little dispersion, very little standard deviations. And if you see the next data sets, you could see that there's a little broadening of the peak, which means that you actually allowed these particular data sets or data which are little bit scattered across the across the mean or the average. We still took those particular data sets, and therefore we have a little flattening or widening of the graph. Everything is fine. Nothing is bad and nothing is good and nothing is standard, but it's just kind of what kind of experiments you are trying to do. A few of the experiments are extremely sensitive, so you need to be extremely tight, and you do not allow anything more than 1 standard deviations to come into our data. But if a few kind of experiments are a little more relaxed and stringent, then we actually allowed a little dispersion to happen, and that is okay. I mean, just understand that different experimental conditions or different experiments has different requirements, and just you are going to follow those requirements and analyze the data. We don't want to be extremely productive and then lose very important data sets, because you just imagine that if you're going for 1 standard deviations, you're going to lose a chunk of large data you're going to throw out of those of your analysis. Now that is okay, but you are going to have lots of data which is actually being lost. We don't want that always. Sometimes those data is very precious. So this all depends upon case to case. So there is nothing like a rule of thumb that you're going to use only 1 or 2 standard deviation. It depends on the particular researcher and a particular set of conditions and for a particular set of experiments. So this is the example I was talking about. Like you could see that there's a little tight peaks, and you could see that there are some example of a broadened peak where there's a large standard deviation. Incidence of diabetes mellitus or obesities. So like this kind of, you could have different kind of an association studies. We are not doing an association studies per se in terms of genomics, but we are just took a data set and trying to find out whether this 2 data actually behaves or moves in the same directions, opposite directions, or there is no relationship between these. Now for that, this particular metric is very important and very helpful. That is Pearson's correlation coefficient. Now Pearson's correlation coefficient can have any values ranging from -1, 0 and +1. Now these are the 3 different avenues, 3 different values which possibly will come from this Pearson's correlation coefficient. If we say a Pearson's correlation coefficient is equal to 1, this means these 2 data, those 2 trends are exactly moving in the same directions. Like for instance, the incidence of cardiovascular disease along with say consumption of say saturated fats. I mean, kind of they had correlated together. Or say for instance, the cigarette smoking and lung cancers, or chewing of tobaccos and oral cancers. So this kind of things actually moves in pretty much in the same direction. So we'll have a correlation coefficient pointing in the same directions. Now sometimes we may not have any relationship between these 2 data sets, and then the correlation coefficient becomes 0. Say for instance, the incidence of the cancers, and someone say wearing a particular dress. We know that of course there is no relationship between these, and if you try to find out Pearson's correlation coefficient between them, we see that actually they don't correlate at all. So P is equal to 0 or rho is equal to 0. This means they don't correlate with each other. Sometimes there's an anti-correlationship where when one data actually goes up, the other goes down. So something like this incidences of say diabetes mellitus or regulation of the blood sugars or how you're maintaining the blood sugar along with say exercises. Now if someone do a lot of exercise, then the blood sugar is going to be better controlled and the blood sugar is going to go down. Now the diabetes mellitus will be better controlled. So you know that one of these is actually being more if exercises are more, then the lifestyle related to diabetes complications is going to go down. So these are moving in little opposite directions. So there the correlation coefficient will be negative, where you know that the two data sets will tend, or the two trends actually moving in an opposite directions. Okay. Now we are going to bring up another very interesting aspect of this data analysis is what we call as the Pearson's correlation coefficient. So what is that? So there are two data sets, and we just want to find the relationship between them. So if suppose we try to find out the association between two different things. So we wanted to see that how the movement of one data is actually correlated to the movement of other data in which direction. So we talk about correlationship between these two data. Say for instance we wanted to find out the correlationship between the incidences of cigarette smoking and that of the lung cancer. So we are trying to find out the correlationship between say sedentary lifestyles and the incidence of diabetes mellitus or obesity. So like this kind of, you could have different kind of an association studies. We are not doing an association studies per se in terms of genomics, but we are just took a data set and trying to find out whether this two data actually behaves or moves in the same directions, opposite directions, or there is no relationship between these. Now for that, this particular metric is very important and very helpful. That is Pearson's correlation coefficient. Now Pearson's correlation coefficient can have any values ranging from -1, 0, and +1. Now these are the three different avenues, three different values which possibly will come from this Pearson's correlation coefficient. If we say a Pearson's correlation coefficient is equal to 1, this means these two data, those two trends are exactly moving in the same directions. Like for instance, the incidence of cardiovascular disease along with say consumption of say saturated fats. I mean kind of they had correlated together. Or say for instance, the cigarette smoking and lung cancers or chewing of tobaccos and oral cancer. So this kind of things actually moves in pretty much in the same direction. So we'll have a correlation coefficient pointing in the same directions. Now sometimes we may not have any relationship between these two data sets, and then the correlation coefficient becomes 0. Say for instance the incidence of the cancers and someone say wearing a particular dress. We know that of course there is no relationship between these, and if you try to find out Pearson's correlation coefficient between them, we see that actually they don't correlate at all. So P is equal to 0 or rho is equal to 0. This means they don't correlate with each other. Sometimes there's an anti-correlationship where when one data actually goes up, the other goes down. So something like this incidences of say diabetes mellitus or regulation of the blood sugars or how you're maintaining the blood sugar along with say exercises. Now if someone do a lot of exercise, then the blood sugar is going to be better controlled and the blood sugar is going to go down. Now the diabetes mellitus will be better controlled. So we know that one of these is actually being more if exercises are more, then the lifestyle related to diabetes complications is going to go down. So these are moving in little opposite directions. So there the correlation coefficient will be negative, where that the two data sets will tend, or the two trends actually moving in an opposite directions. So this is a heat map representation of the same thing. If you see in the first profile, wherever there is in blue and wherever there is in yellow, you could see how the relationship. Now let's see in the second one. You could see the relationship between the second one, that wherever there is in blue, then there is in yellow in another one. So there are actually two sets of the data, the upper panel and the lower panel. You are trying to mix these two data sets and trying to see how these two data behaves. Like that's the whole experiment that was being done over here. Now in the middle row, if you see, wherever there is a blue, there is a yellow, and wherever there is a yellow, there is a blue. So there is not too much of correlationships in the third panel, right? But in the second panel, what you see is that this is very random. You can see there is a blue and there is a yellow and there is a yellow. So this is kind of a little unpredictable. So in that kind of scenario, the middle panel actually shows that they are not correlated because we cannot predict them. The two data sets are not being predicted. Their trends are not being predicted. So the middle panel is a clear example of a scenario where the two data sets are not been correlated with each other, because the blue and the yellow patterns doesn't follow the same trend. They are not regular, they are just very random and chaotic. But in the lowest panel, if you see, they are anti-correlated. I was talking a little earlier, wherever there is a blue, there is a yellow, and wherever there is a yellow, there is a blue. This means one of the data sets is showing an upregulation, the other one is showing a downregulation. So this is an anti-correlated, and therefore you could see from the data with the R values, this -1, this means they are anti-correlated. R is equal to 0, this means there is no correlation between these two. And the first panel actually shows a value equal to 1, which shows that wherever there is a blue, there is a yellow. Wherever there is a yellow, there is a blue. This means the two data sets are actually moving in the same directions. And therefore you could see, understand with this couple of different examples, that how the Pearson's correlation coefficient is so vital in interpreting the data, in addition to the using the systems biology tools to find out the trends in the data sets. So these are something which is very important to understand, that they could be correlated because of some independent reasons. They may not be drivers of each other, but at the same time simply the two data sets may be correlated in the same directions. So just a mere correlation doesn't mean that one is cause and another is the effect, or one is the mechanism of that. So next is that when you have a data sets, like for instance the data sets which comes from an RNA-seq data or from an expression array data, then we make the heat maps, and then once we make the heat maps, then we actually ask the very simple questions. Is there a pattern in the data sets? And if there are a pattern in the data sets, what sorts of data actually behaves together? What sorts of data looks differently? I'll give an example. Suppose you take your cells, grow them in tissue culture systems, and then stimulate them with a drug, say for instance paclitaxel or some other anti-cancer drugs. We know that a few of the genes will be activated once this drug is actually been added, and then the cell eventually is going to go and show in signs of apoptosis or cell death, because that's the whole purpose of this anti-cancer drug, right? Now when we look into the data and ask a very simple question, okay, when the cell is actually dying, what sorts of the genes or what sets of the genes are behaving together, and what sets of the genes are actually being clustered together? So if you look into a gene expression profile of SS cells which is been treated with an anti-cancer drugs, and then look into the data and analyze it, then you will see that the genes which is related to apoptotic pathways are all being clustered and tightly joined together, and the genes which has been related to some other pathways are actually, say for instance phagocytosis, or say for instance autosomes or autophages are going to be all clustered together. So just by looking into these clusters, we can actually say that these sets of the data actually admit together. Is what exactly is the basis of the clustering algorithms, is that they try to take the distance of, calculate the distances with each of the data in the XY coordinates. If we plot them in a two-dimensional XY Cartesian coordinates, then we will have the X coordinates and the Y coordinates, and then each of these X and Y coordinates can have their own XY values, and then what you are going to do for each of the data set is trying to find out the distance between these two data points in the two-dimensional Cartesian coordinates. So we know the standard formula that y minus x is the one in which you actually calculate it. There's a formula for that, right? And then just by using this formula, applying this formula, you're trying to find out what is the distance between these two data points. If the distance is least to the minimum, then we say that okay, these two data are possibly related to each other, so they should be clustered together. That's the fundamental way in which the hierarchical clustering algorithm actually works too. They calculating the distance between. So I'm just giving an example of how to cluster the data sets. So for instance, we have two persons, couple of persons, and they actually been using two different variables like x1 and x2. So now looking into the data sets, if someone throws a data set like that, it's very easy for us to actually look into this and then tell that okay, what sets of the data has actually been clustered together and what sets of the data has actually been separated out from each other. Now whenever we are doing that visually or mentally, remember what we are actually doing in our subconscious mind is we are trying to find out the distance between these two data sets, and that's exactly what the clustering algorithms do, is that when the data sets actually contains a large number of data which is been scattered separately, we are trying to find out that what is the distance between one data and another data, and where the distance is minimum, we say that okay, these two data tends to be together. So this is purely on the basis of finding out the distance between these two data points in say two-dimensional or three-dimensional space is what exactly is the basis of the clustering algorithms, is that they try to take the distance of, calculate the distances with each of the data in the XY coordinates. If you plot them in a two-dimensional XY Cartesian coordinates, then you will have the X coordinates and the Y coordinates, and then each of these X and Y coordinates can have their own XY values, and then what you are going to do for each of the data set is trying to find out the distance between these two data points in the two-dimensional Cartesian coordinates. So we know the standard formula that y minus x is the one in which you actually calculate it. There's a formula for that, right? And then just by using this formula, applying this formula, you're trying to find out what is the distance between these two data points. If the distance is least or the minimum, then we say that okay, these two data are possibly related to each other, so they should be clustered together. That's the fundamental way in which the hierarchical clustering algorithm actually works too. We're calculating the distance between them. So this is again the example which I was talking a little back, that the A, B and C data sets, if you see over there, and D, E and C, B and D data sets, if you see the two sets of the data, D and A, they are clustered together. So another set of the data, C, B and E are actually clustered together. So it is very easy for us to understand and interpret the data and tell that okay, A is related to D more than A is related to B or C or E, and vice versa. That B's relationship with C and E is more as compared to D or A. And the basis of that is that the distance calculations and how the distance between these data which are clustered together is least or less as compared to the distance between the data which are clustered differently. So this is the way what I was talking about, the distance formula. You could see over there that x2 minus x1 squares plus y2 minus y1 square root of that. That's a standard formula which people always use it to calculate the distance between the 2 points in a space, and that's exactly what is actually being utilized in this. And that is what I was talking about a little back, that we calculate the distance, and if the distance is minimum, then we say that okay, these points are actually related to each other, so let's cluster them together. So this is another example where we actually been showing the same thing, but we're actually calculating the distance using the distance formula. This is actual calculations. We could actually play around with these data sets and feed the different coordinates, the x and y coordinates, and then just try to calculate the distance using that distance formula. And you would see by yourselves that where the distances are actually more, then these two data sets tends to be far away from each other. This is just a very good tutorial for you to try in home. Just try with the different values of x and y, and you would see that there is a very good correlation between the data which are clustered together and the distances between them. So when the data are clustered together, the distances between them is minimum as compared to the data which are actually situated far away, and the distances between them is large or lots. So this is the heat map, which is actually final interpretation. So when you look at a heat map, what exactly that has happened now, that has given rise to a heat map, is actually calculations of hundreds and thousands of the distances between the data sets. Each of the data sets is actually represented by their expression values. We are trying to see that what are the distances between two expression values. And after doing that, we are trying to make the data sets and look into heat map formats where you could see that the values or the data which are clustered together are given a particular color code as compared to that of the data which are actually separated away from each other. And our experience actually tells us, and it's very easy to interpret this data, that those values of expressions of those sets of the genes which actually regulate similar kind of pathways are all being clustered together in one cluster. In other words, their expression values are actually very related to each other. As an example, giving you an example of the paclitaxel treatment with the cancer cells, you could see that all the sets of the genes related to apoptotic pathways like BCL2, BAX, BAD, BEAM, all of these are all being clustered together because they all actually direct one common pathway, and that is the apoptotic pathways. But that will be far away from, say for instance, immune pathways, because immune systems may not be playing a very active role in initiating the apoptosis when you are going to treat a cells with paclitaxels. So you could see that just by looking into those data sets clustered together, you could find out the correlations. Sometimes we could also tell that okay, all of the sets of the genes actually are coming on operational within one particular pathway. So that's the value and that's the power of this kind of an algorithms and this kind of an analysis. So these are the few of the text which you are encouraged to go back and then read, and then actually kind of enrich your knowledge and information about this particular area. So this is just a reference, a couple of references will be also there in the e-text. So feel free to go and read of them. And it is not very hard, but just spend some time and try to understand the crux of this entire thing. So coming back again, just kind of a summary that the goal of this clustering is to subdivide the items in such a manner that the similar items actually fall together. That's what I was just giving you with different examples. Now so far you have seen a method of clustering which is very commonly used in biology, that is known as the hierarchical clustering. Of course, there are other kinds of clustering, because most of the time hierarchical clustering is fine, okay, but many times we need to do different kind of clusterings because to get out different information out of the data sets. And then there are additional means of clustering, like K means of clustering is another very smarter ways or smarter algorithms to find out how the data sets actually been clustered together or whether they have been separated out from each other. Now this is a very this is a table. It actually shows you the list of the different formulas which are actually been used to calculate the distance. See, that's what I was trying to tell you, that all the things clustering algorithms works, the underlying mechanism behind them, which is operational, is actually calculating the distances between these two data points. And more the distance, the more separated from each other, and vice versa. There are different formulas to calculate the distance, like Manhattan's distance, Euclidean distance, Mahalanobis distance. But these are the different advantages and disadvantages of using these mathematical formulas at different data sets. So it's not universal, so it all depends upon the researcher, what kind of information they want to dig out of the data sets. But these are the different equations which is actually been available to us, and you're going to use these equations one or the other depending upon what kind of problem you are addressing and how you are going to interpret the particular data. So this is the list of these different formulas which is being used to calculate the distance is being shown in this particular table. Feel free to see them, and if you minutely see them, they are all calculating the distances between the two data points. That's the whole philosophy behind this clustering algorithms. So this is the one which I was talking about, that hierarchical clustering and partitionings. So this is the similar kind of thing that most of the clustering algorithms work, is that how they partition the data sets and find out the patterns between them. Okay, so there is another kind of cluster which you need to be aware of, and that is what you call as the K-means clusterings. And this is a centroid-based clustering. I'm going to talk a little bit about this, not too much, because it's mathematically a little exhaustive if you talk about too much, but that's not the scope of this module. But you are going to just see into what exactly this K-means clustering. So it's a centroid-based clustering. I'm going to try to explain to you in simple terms. Take an example of, say, this is a data set in a two-dimensional Euclidean space, and this data is actually been separated out in the different regions in the space. So what you do is that you just draw arbitrary partitions between these data sets, and once you make an artificial partition, this is a well self-defined partition. There is nothing like someone is asking you to partition at this particular point, but this is a self-defined partition. You just can user-defined. You just define yourselves and partition the data into different clusters. And then if you take a particular sets of the data and try to find out the centroid of that particular data sets, and then you calculate the centroid of, say, data sets A, B, and C, and then you have another sets of the data and just partition them because they are actually together, and then you find the centroid of that particular data set, so centroid of, say, E, F, and G, and then we try to find out the centroid of another data sets, and then we look into those centroids of all the centroids of these data sets which we have partitioned. And then those centroids, where the distance between these two centroids is actually minimum, we are trying to cluster those centroids together and put them into one cluster. So initially they are separated out, right? So we artificially draw some partitions, and individual data in the partitions are actually been taken, and a centroid or a central point is actually been selected. Similarly, there is another set of the data points, and their central points of the centroid is actually selected. And like that, we have bunches of different centroids for the different sets of the data points, right? Now we are now calculating that which two centroids are closest to each other. So which two center of the data points for the sets of the data are actually been clustered together or closer to each other. Now if they are closest to each other, the minimum distance one, we pull them together and make one big giant cluster, and then actually say that, okay, these two centroids are all together because the distance is minimum, and therefore we put them into one cluster. So essentially what we are trying to do, what we are achieving in this particular K-means clustering is that we are reducing the number of centroids and we are bringing them as a minimum as possible till you reach to that stage where we cannot have an additional reduction in the number of the centroids. So you start with n number of centroids and then you actually reduce them slowly and slowly once you cluster them together into one group to a point where you cannot further reduce them. And that is the whole purpose of this K-means clustering, is that this is a centroid-based clustering which is aimed in trying to find the centroid of the data sets and then clubbing them together. This is again the purpose is the same, trying to find the pattern in the data sets and trying to ask a very simple questions. Which set of the data actually behaves together, which set of the data is actually being separated or behaving in a different way. just to find out the pattern and a trend in a particular data sets. So this is what we are talking about, the centroid based methods, and then the stars are actually the centroids of these data sets and dots are actually the data, and this is exactly what you actually been doing. So these are very arbitrary ways of partitioning the data, but then it works fantastic when you look into the big data set. That's exactly how the algorithm actually really works in the code of the computer. Okay, so there are certain plus and minuses of these clustering algorithms. Is that most of the clustering algorithms sometimes can miss out some important clusters, or sometimes you may have something which you look like they're correlated, but just because since this is so much mathematically driven, you might lose out those data points and then you might put them into different clusters. So my recommendation to you is that just don't blindly go into these clustering algorithms and let them to decide everything of your data sets. Be focused onto those data sets and look manually, individually, eyeballing on them, and then see that in terms of for us, what's more important is that we need to find out the biological relevance of this data, the relevance in biological terms. We just don't have to cluster the data for the heck of clustering. We try to cluster the data and then try to ask a very simple question: is that does this makes any sense? Does it dictates a particular phenotype? Does it explains a particular experimental observations? And therefore, those fine tuning, depending upon what exactly is we are expecting, what is the phenotype, how the data looks like, whether that is closely representing the phenotype, is the additional responsibilities of the researcher to look into, and then further monitor the data sets. Don't just leave everything for the clustering algorithms to do that one, because in that case you might lose some important clusters, or you might lose some important associations. So that's the word of caution. So there are few requirements of a good clustering programs or good clustering methods. And we can talk about this a lot, but again, this is something which you can always go back and read into any standard bioinformatics or systems biology books. Just wanted to talk a few things is that many times when you talk about the data sets, you talk about the dimensionality. So you have one variables, one dimensions, two variables, two dimensions. The dimensionality in a biological experiment actually increases dramatically if you are analyzing different data sets and the different conditions and the different treatments at different time points. So you dramatically increase the dimensionality. So your clustering algorithm should be able to cope up with those dimensionality, the higher dimensions, and it should not collapse. So mathematically, it should be operational in a multiple in a hyperspace kind of a thing, where the data sets, if the dimension increases, then those kind of extra spaces pops in, and your equation should be able to support those kind of extra spaces, because this is very common for biological experiments or to have multiple variables and multiple dimensions. So your clustering algorithm should be able to support that, and most important, you should be aware of that kind of variability happening in the equation. So that is the reason why I say that just have in caution your individual input into those data sets, and then don't let everything for the algorithms to decide on it. So the similarities and the dissimilarities are actually been represented in the data sets. Another very important thing about systems biology is that so far you have been talking more into the data analysis point of view, and that is okay, and that is very good for systems biology. I'm going to talk a little bit in the concluding minutes about why should we study systems biology in that rigorous way, and what are the advantages and disadvantages of that. As I mentioned in the beginning of this lecture, is that in systems biology the convention is a little different, where you start with a large data set and trying to find out the pattern with the data sets, rather than a conventional experiment where you start with experiments and get the data and then interpret it. So with systems biology, it has been assumed that you already have a big data sets, say a data set from a mass spectrometry experiment, or a data set from an RNA-seq experiment, or a data set from genome-wide association studies. So this kind of large data sets are very good candidates to start with the systems biology approach. The next thing about systems biology is that you need to understand how the biological systems actually works, rather than a conventional experiment where you start with experiments and get the data and then interpret it. So with systems biology, it has been assumed that you already have a big data sets, say a data set from a mass spectrometry experiment, or a data set from an RNA-seq experiment, or a data set from genome-wide association studies. So this kind of large data sets are very good candidates to start with the systems biology approach. The next thing about systems biology is that you need to understand how the biological systems actually works. Majority of the biological systems, what we know for, are actually works in a way which is little different than the conventional wisdom what we have. So in biological systems, there is something called the correlationship between the systems, and there is something called the transfer of the information from one system to another systems. Now, biological systems, if you understand that, they are actually the open systems. What does that mean? That means that these systems can actually communicate with the external atmosphere and environment and can get the information out of that. So we should be aware of that. The systems biology has to deal or always deals with an open systems. Now, another thing about the biological systems is that majority of the biological systems falls under a pattern which is not or which cannot be directly interpreted with the help of well-defined equations. Or in other words, many of the things in the biological systems doesn't follow a continuum principle or a continuum law. So if we have certain sets of experiments which is going on, or certain sets of data points which is actually over there, just like looking into that particular set of data points, you may not be able to interpret the whole systems completely. And that is why exactly systems biology is helpful. So we need an integrated approach, and we need a more coherent approach to look into the data points and then try to understand and ask a simple question: how does the whole systems behaves? Another very common feature of the biological systems is that when the biological systems, if you kind of do any kind of perturbations in the biological systems, this system has lots of plasticity, and because of that, it is going to resist that particular perturbations, but beyond a particular threshold. And if the perturbations in the biological systems crosses that threshold, then it will be set to a new or a different kind of a platform, and that new platforms is different from these old platforms. So biological systems actually operates in a way. Example is that if you have a transcriptional network, say particular sets of the genes, and then you take those particular cell and then actually stimulate them with a different stimulants, say cytokines or hormones or a drug, and then you will see that this system actually adapts to a different kind of transcriptome signatures once you do that one. So we say that there is a transition in the systems, and that is also one of the very important features of the systems biology, is that tries to find out how the systems are, which way the system is actually making transitions from one state to another state. And we can say that this transition is actually the underlying causes of different kind of pathologies associated in the biological systems, like cancer, like disease states, and so forth. Okay, so once we have in biological systems and once we have a set of the data, now another very important thing in systems biology is building networks. Networks is a way of connectivity between these different into the systems. So when we say networks, we say that the network of the genes. Now, even though we didn't use this word actually actively, but just remember that whatever we have been talking so far, underlying principles, everything, or underlying mechanism, everything, what we're trying to do is actually we are trying to build the networks, we are trying to integrate the networks. So we said that the networks of the genes which are related to apoptosis, the network of the genes which are related to the DNA replications, the networks of the genes which are related to a particular biological process. So network is actually connectivity, and this is actually one of the very important strong aspects of the systems biology, is that it is very efficient in making the networks. And with this, but seeing these networks, seeing this connectivity, we can say that how the two systems are actually even related to each other. Now, in a different aspect, let me give an example. Like for instance, you take a particular gene, your favorite gene, say for instance the gene X, so say P53. Now, in a different aspect, let me give an example. Like for instance, you take a particular gene, your favorite gene, say for instance the gene X, so say P53, and then majority of the time we kind of try to work on P53 in either over expressing that one or say knock it down. And when you knock it down, P53, and then after knocking down, if you do an expression or a transcriptome assay of that particular systems, then you see that not just P53, but there are a lot of different genes which gets altered. Now, if you look in the sets of the genes which get altered, the most one would be the one which is closely related to P53, the first neighbor, we tell that. So the first neighbors of the P53 will be altered. But if you see this particular network of the genes which has been altered and build this network and see this network, you will see that many genes which are farther away from the P53 pathways are also been altered, because they are actually not by direct way, but with the help of a wire mechanism, so with the help of an additional mechanisms. When one gene actually alters one network, and then that network actually perturbs the next network, and it actually goes in that way. So it builds up in a way a very complex system. So all what is very important is that now in the systems biology approach, you should be aware of that just if you take one particular gene and knock it down or knock it out, which you very commonly do in biological experiments, be prepared to understand or face the consequences of that, because the whole systems may actually undergo dramatic changes, because everything is actually been connected to each other with the help of connectivities, and these connectivities are very important to understand the phenotype of the systems. So there is something called the gene enrichments. So there is different pathways to do that one. So there are different algorithms, different softwares. Many of them are freeware, so you can actually use them. Say for instance, the David's pathway for analysis of the gene expression. Say for instance, you do an experiment where you took a cancerous cell, you took a normal cells, or you took a disease cell and you can control healthy cells. And then you do a transcriptome signature of these two different cohorts, the normal versus the altered one or the normal one, and then you got a particular sets of the genes which are differentially expressed, right? That's the standard thing. Now what you do is that you take these gene sets which are differentially altered with a specific p-values and a specific cutoff. Say for instance, we say that if the alteration is more than one fold, we are going to take that. You may be aware of that there are different researchers using different cutoff values. Sometimes they may say that, okay, if the fold changes is more than 2.5 fold, I'm even more stringent, then only I'm going to say that, okay, this is differentially altered. So now whatever is your cutoff, whatever is the threshold, whatever is the bar which you kind of selected, that's researcher specific. But once you set up that bar, then you have a set of the genes which are differentially expressed. Now what you do with these sets of the genes is that we run them through different kind of programs like ingenuity pathways or David pathways, and by running them through these programs we actually try to find out what exactly these sets of the genes which are differentially altered mean to us. And you could see that they, most of the time, they actually enrich to a particular pathways. As I give an example a few minutes back, that if you take paclitaxel, then you will see that all the genes related to apoptotic pathways or cell survivals will actually be clustered together. All the cells which is related to proliferations will actually be clustered together. So you could see that in these David pathways or ingenuity pathways, the different pathways get actually enriched. And those pathways which get enriched is because the sets of the genes which is related to each other in a particular pathways are all clustered together, and their alterations actually happens together. So all the apoptotic pathway genes, BCL2, BAX and so forth, where their expression is going to change dramatically, and all of those genes will actually be together, clubbed together, and you could clearly see that an apoptotic pathway actually gets enriched. So this kind of enrichment of the pathways actually is very common once you take the data sets and then actually feed them through this kind of pathway analysis algorithms like ingenuity pathways or David pathways or cake pathways and so forth. Okay, so just to come back to almost the end of this discussions today, remember the systems biology is an integrated approach rather than looking at the systems in a broken splitted portions. We are trying to finally integrate the whole system as a whole. It doesn't mean that in systems biology you need to start with a big data or big organ or big tissues, but at the same time it is very important that we need to consider that all of the data sets can be correlated together, and the interpretation is only when we actually join them together into one platforms. Now this is very important that in systems biology you should be prepared to handle big and large data sets, and therefore the different kind of statistical and mathematical tools that comes in is very helpful. One of the tools or few of the ways in which we can look into the pattern in the data sets is what I discussed, is that clustering methods and then building the networks. So when you build the networks, exactly what you're doing is that we can make models out of this, and this building the models and the networks actually help the researcher to predict how the system is going to behave in the later course of time. It's just like in the stock markets, when you actually look into the fluctuations of a few stocks, and if you just follow them for a particular point of time, regularly and monitoring their fluctuations, then you can actually predict them, that how this particular firm or company is going to work or perform in the next 2 months or next 1 month or let's say next 1 week, because we have followed a pattern of that system. Similarly, when we take into systems biology, we can actually look into the systems and then see how the systems behave with the help of different kind of tools and techniques, and then from there we are going to build a model, and that's an operational model. This model is futuristic because this model is going to tell how exactly the system is going to behave in a later course of time, what is the trajectory of the systems. And these are very important because just by building models and just by doing some kind of computational simulations, you can actually predict the behavior of a complex systems. But remember that is just the ease of it, but at the end of the day you need a wet lab validations of all of these predictions and then come to a meaningful conclusions. And therefore most of the time in clinical studies we have something called the discovery cohorts, where you use this kind of fancy tools techniques to make some novel discoveries out of it. And then we have a validation cohorts where we take all the discoveries which we have from the discovery cohorts and try to understand and ask the questions that all of our lists, all of our hits, all of our predictions, how true or how much they actually been holding into a real scenario. So now you have these models. Take this model and then translate it to an say clinical samples or clinical cohorts, and then say that okay, this model has been actually should be predicted. How much predictable is it in an actual clinical scenarios? And that is what is called as validation cohorts. Taking the validation cohorts, we can actually use this data points and do the validations. Now, if that happens, that your validation is nearly good, then you can be very sure that this model is very robust and it can actually predict the behavior of the systems. And if it doesn't, then you definitely need to go back and tweak into the parameters and then see that how good your model is in order to predict the actual real life scenarios, even without doing any experiments. And that's the advantages of doing the systems biology approach. So systems biology approach is a very integrated approach. It is an approach where we look into the whole system together rather than looking into parts. It starts with analysis of the large data sets, trying to find the patterns and correlations in the data sets, trying to find out the networks and the connectivity within the data sets, and trying to find out and build the models within the data sets. Now all of these actually together are the domains of the systems biology. It's a rapidly expanding field, and it is even more expanding and even more prominence because with the development of the technologies that actually helps to fuel the systems biology approach, like NGS approaches and then mass spectrometry. You want imagine that even 10 years back, the next gen sequencing was kind of prelim, and then not every labs or not every institutions could utilize that. And even if they could afford it, the data interpretation algorithms were not that robust, and they was not able to go anywhere from there. But today, standing today in 2018, we have these platforms for the data analysis which are extremely robust and very user friendly. Now we can look into the large data sets much more with much more confidence, and actually can interpret the data. And this kind of wet lab tools and techniques like next gen sequencing and massive parallel sequencing has enriched this particular area of systems biology, and together they are just like hands and gloves, enriching each other. And systems biology is a way in which you interpret these large complex data sets and find out a meaningful pattern out of it. And most of the time, once you do in systems biology kind of an approach, you will see that you are very close to kind of predicting exactly the real life scenarios. This kind of problems actually happens when people kind of was trying to do a single cell based experiments and take that particular data and trying to go it to the clinics, and they found that most of the time this really didn't work. Majority of the discoveries actually fails. They couldn't even reach to the phase 2 trials because majority of the data points seems that even though they are fantastic in a tissue culture model, but once you translate it to a patient or to an actual clinical scenario, this data point actually loses all its charm. It doesn't interpret anything because the approach was different. Now we are even more knowledgeable and more enlightened, and we can actually know how the data behaves, and using all the tools and techniques of systems biology, we are in a much more and better prediction in a predictive mode, or we can have a much more higher predictive power to predict about the behavior of the systems. So thank you for your attention, and I'm sure that you will have a couple of questions. Feel free to go back to the references which has been listed over there and listen to a couple of other videos and tutorials related to systems biology, and I'm pretty sure that you will enjoy reading them, and you will have a different way of now looking into biological systems with much more robust way, and trust me, is going to give you a better interpretations than if you just do in a single cell biology based way. So thank you for your attention.